Hello everyone. In this video we will see how you can use Prokka for genome annotation. After that we will look at the generated files and analyze them. So first of all we need to create a new environment where we can install the tool Prokka. For that go to your terminal and type. So here I have written conda create then double dash name and Prokka. So I am telling the terminal to create a new conda environment with the name Prokka. And in my computer, I already have installed these four channels. And for Prokka particularly, you will be needing Bioconda. So make sure that you have the channel installed. Now you have to press Y to proceed. So the environment has been created. I will clear the screen and type conda activate Prokka. Now you see that it has changed from base to Prokka, meaning that the environment has been activated. Now the next thing is to install the tool Prokka within this environment. So for that, go to your browser and type Prokka installation using Anaconda. So Anaconda basically helps you to manage packages like Prokka and other tools that you will be using at the time of your bioinformatics analysis. This will make your workflow way easier than going to the browser and downloading it yourself. So open the link and here you can see that to install this package that is Prokka, you need to run either this command or the one below it. So I will be copying the first command and pasting it within my terminal. So now the environment Prokka is activated and I am installing the tool Prokka within that environment. As I mentioned earlier, it needs Bioconda, so make sure that you have the channel Bioconda for running Prokka. Press Y and click enter. So the required dependencies are being installed now. Now the installation of Prokka has been completed and we can check if the Prokka is installed correctly or not by typing Prokka-H. So here you can see that it has mentioned the Prokka version number and every option that it gives for you to run any file. Now that we have the Prokka installed, we need to download the genome file that we want to annotate using Prokka. So for that, go to NCBI and here select genome. Then type the name of the species that you want to download. For example, I am taking Bacillus subtilis subspecies subtilis. So here I have found 165 genomes, but this contain from contig level to the complete level. Prokka can be used for any of these levels because Prokka can work on the minimal level, which is the contig. If you want to only download the complete genome, you can just drag it here to complete, and from 165 it has changed to 112 genomes. As an example, I will be taking the first genome. So click on these three dots and go to download. So we want to download the FASTA file and only refsec one. You can change the name of the file here if you want. Suppose I give it the name subtilis and click download. Our download has been completed. Now we need to extract the contents present in this zip file. For that, go to the folder where you have downloaded the file. So I will be going to downloads, and here is the file that I downloaded. Then I will extract the contents. So in the subtilis folder, I will go to NCBI dataset, then data, then GCF, and here is the .fna file, which is the nucleotide file for the genome that you just downloaded. So this file will be used as the input for Prokka. Now that we have our input file, we will again go to our terminal. So make sure that the environment that you just created with the name Prokka is activated in your terminal, which can be confirmed by this name in the bracket. Now to start the run, we will first type Prokka to initiate the Prokka tool. Then we can specify the CPUs. I am having 14 CPUs in my system. So if I use 10, it will be sufficient. If you increase the number of CPUs, the runtime will be significantly shortened. So if your system contains a higher number of CPUs, you can change them accordingly. Next, we can specify the kingdom. Because I am annotating a bacterial genome, I can specify the bacteria kingdom here. So this tells Prokka to search only in the bacterial kingdom genomes and not go to other kingdoms. It will save your time. Then you can add the locus tag if you want. So locus tag is basically every gene that it annotates will be given a tag, and the initials can be defined by the user. For example, I am annotating bacillus subtilis genome here, so I can just add subtilis at the starting of each tag. After that, you can add genes section to your output file. Obviously, it adds the genes to your file and nothing else. Then you have to specify the output directory. So suppose I want to save the file in downloads folder with the name annotated subtilis. So a folder will be created in the downloads folder where the results will be stored. And last, you have to specify the input file that we have seen here. So specify the path where your file is present, and the path is this one. So type downloads, then subtilis, NCBI dataset, data, GCF, and in this GCF folder we have the file, this one, and click on enter. So the Prokka has started running now, and you don't need to add every of these options. You can skip out on any of them if you don't need them, or you can add additional options if you want, that we just saw earlier while we typed Prokka-H. So it will take around 1 to 2 minutes depending on the genome size. It actually took shorter than a minute to annotate the genome that I just gave it. So depending on the genome size, it can vary from less than a minute to around 1 to 2 minutes. And it also depends on the number of CPUs that you are giving it. And these are the output files that are generated. We can now go to the downloads folder where we have created this annotated subtilis folder and look at the results file there as well. So go to downloads, and here is the folder that we just created. These are all the files that are generated by Prokka. A total of 12 files are generated by Prokka. Some of these will be used as the input for further downstream analysis like KEGG analysis, etc. We can take a look at some of the important ones. For example, if I open .faa file, you will see all the genes that are annotated by Prokka and their respective amino acid sequences. Additionally, as we set the locus tag to subtilis, we are seeing subtilis at the start of each gene sequence. While if I go to the .ffn file, it contains the nucleotide sequence of each gene that the Prokka annotated. Now, based on your downstream analysis, if you want the amino acid sequence, you can go for the .faa file, and if you want the nucleotide sequence to be analyzed, you can go for the .ffn file. However, the problem emerges when you want to count the total number of genes annotated by Prokka. Counting each of the gene one by one will take a lot of time. That's where command line options come into play. We can use the grep command for this task. Just go to your terminal and type grep, which is the command name, then -c, which tells the command to count. Next, you need to specify what you want to be counted. In this case, we will be counting this character. So I will put this character in single inverted commas. And last, you need to specify the input file path, which is downloads annotated subtilis, and here this is the .ffn file, and press enter. So there are a total of 4332 genes that are annotated by Prokka. So it makes your work a lot easier, because if you have started to count all of these genes one by one, it would have been impossible. Just one more thing, you don't need to activate the Prokka environment for this task, because the grep command is an inbuilt command in the Linux terminal. So that is all for today's video, and stay tuned for more bioinformatics stuff. Thank you.